Okay, let's start. Okay, good morning, and we are going to start our fourth session. So our fourth session, it has two parts. First part is protein-protein interaction, and the second part we will start on few methods of proteomic technologies that are used in studying different proteins, the different proteins processes or modification interactions and other processes in the cell. So the first part is about protein-protein interactions. So for this first part, the session learning outcomes you will be able to define the types of protein-protein interactions, explain the structural and chemical properties of protein-protein association interfaces, describe the characteristics of each type of protein-protein interactions, and explain the methods used for identifying protein-protein interactions in vivo and in vitro and in silico. Let me get in your colleagues. I have to go back to the. Okay. So go back to the session. So what are the common terminologies that we are going to come through in this session? So what is binding free energy? Since we are speaking about protein-protein interactions, then there is definitely an energy production and energy integration within the process of binding or going into chemical reaction between two polypeptide chains. So binding free energy is defined as the free energy differences between the bound and completely unbound state of a polypeptide chain. So it's a function of the amount of surface of each protein. It's according to the surface area of the protein or polypeptides that got into interaction with each others. So usually this part, this surface area is considered hidden from the solvents within the cell. So because they are already involved in complex formation or in an interaction between the two polypeptides. So there is a production of energy, and that's why we call it binding free energy, and it is the difference between bound and completely unbound state of a polypeptide. Okay. So interfaces. This is a term that defines the interacting residues that belong to two polypeptides. And usually within this interface, there are a number of residues, amino acid residues that get into chemical reaction between the side chains of the two of the amino acids that constitute the two polypeptides. There are other amino acids within this interface, but they are not part of this reaction. So the interface consists of those within the reaction that leads to the interaction between a protein and protein, and those is part of this vicinity, but they are not part of the reaction. So an interface is where the two polypeptides, or maybe more than two peptides, they come into an interaction and form a protein complex. So what's a protein complex? Is a group of polypeptide chains linked by non-covalent protein-protein interactions. And we already before in the last sessions, the last session, we already defined what's non-covalent interactions, like hydrogen bonding, van der Waals interactions, and we spoke also about other hydrophobic and hydrophilic interactions. So these are all considered non-covalent. So usually these protein-protein interactions is due to this non-covalent bonding. So what's a protein-protein interactions? These are defined as physical interactions between two or more proteins that occur within a cell as well as between cells. So these interactions, because we are talking about proteins and polypeptide chains, cell membranes are also composed of high percentage of the cell membranes composed of protein. So an interaction between a cell and the other is considered a type of protein-protein interactions. So these interactions leads to the formation of large complex protein complexes. And these protein complexes, they have a specific function within the cell. So some of these proteins, polypeptides, once they are translated or synthesized and modified, then the third step, they tend to form their complexes by interacting with other proteins. This is part of their functional conformation, to be attached to other proteins so that they can act and they can have their function within the cell. So interaction among proteins depends upon various factors within the protein itself. That is mean its sequence, its tertiary or secondary structure, its other associated factors, maybe presence of certain metals, the presence of water, the pH of the cell. All these affect the protein interactions. And even its three-dimensional shape or tertiary shape of the protein, it affects its interaction with other proteins. So these protein-protein interactions are hydrophobic. They have a hydrophobic effect. So most of the interactions depends on, not, as we mentioned before, on non-covalent reactions or non-covalent bonds. And these are like hydrogen bonds, electrostatic interactions. These are the most important types of interactions, the important connections or binding between proteins that lead to protein-protein interaction. In some cases, in some interactions, covalent bonds like disulfide bonds can take place, especially between amino acids like cysteine, if it is involved within the interface of the protein-protein interaction. So many of these protein-protein interactions, it's not only hydrogen bonds or electrostatic. Maybe it's a conformational fitting. That means it has it's one protein can fit within the cleft of another protein because of its conformation of three-dimensional structure. It forms like areas where other proteins can fit without any hydrostatic or hydrogen bonds. So it's like a key and lock interaction or fitting together because of their conformational structure. So we have This interaction, they have different functions, and the determination of this function depends on the composition of the interface, its affinity to each other, the proteins to each other, and this lead to um, different types of protein interactions. We are going to dive more on these types, but there are two types, main types, that depends on the binding affinity of these proteins together, and this is permanent or transient type of interaction. We are going to define these terminologies as we go on into this session, and we have more types of these protein interactions depending on different other characteristics. The protein interaction as a complex, they have different functions and crucial functions within the cell. They could be part of the signal transduction, and this is an important process within the cell that facilitate the pathways of different enzymes production, different biochemical activities within the cell, in the translation process, in the replication of the DNA process, and even in the whole cell cycle, the different phases of the cell cycle. The protein interactions, it's a crucial process for these processes. So why are we studying the protein interactions? First, if you want to study any protein, you have to know its function. To know its function, you need to know what's binding partners, because proteins, most of these proteins as individual polypeptide chain might not be functional unless they go into interaction with other proteins. So to know the protein function, you need to know its binding partners. And the other way, it's the other reason why we study this interaction, protein interactions, to define the cellular pathways, because all this again depend on protein interactions. Even if you know the function of this complex or this protein complex or these interactions, you need to know how it's dynamic, how it process its function. Is it continuous function? Is it transient? Is it permanent? Is it once and in a specific process? So you need to study its dynamic, and these studies are very crucial, because nowadays there is a lot of studies about production of proteins that are therapeutic proteins, but you need to understand the physical and chemical and interaction of these proteins within the cells, so that you proceed in the process of, for example, in drug designs. It's drug design. It's not only for human. It could be for environment. It could be for plant. It could be for animal. So we are making use of the function of these proteins to define the need of the different organisms for certain therapeutic agents, like therapeutic proteins that are that could lead to a better productivity, or curing certain diseases, or reducing mutations within the cell, or production of resistant plants, and so on. So you need to study this protein interaction in vivo and in vitro, and we're going to speak about the different processes within in vivo and in vitro. and their importance in drug discovery. So knowledge of this pathway, its length, its dynamics is very useful for defining the side effects also of if you produce any drug, what's its side effect, how it interacts with um, other proteins, other chemicals, maybe metals, and so on. So these are very important to study. That's why it's very important to study the protein-protein interactions. Okay. So what's the protein-protein interface? Protein, as I mentioned, proteins or polypeptide chains, they interact through their surfaces. And as we talked before about the different levels of structures of a protein, once it is synthesized, they have the primary level, the secondary level, which is the alpha helix, the beta sheets, and their tertiary structure, their quaternary structure. Their quaternary structure, it represents what we are talking about today, about the protein-protein interface. So usually the protein, once it takes its conformation, then it exposes its R groups or the side chain groups to the surface of the polypeptide chain, and these R groups or the side chains are very active and reactive component of the amino acids. So they tend to react with other proteins by different non-covalent reactions. So once they go into these reactions and combine with other proteins, the surface that is part of these reactions, we call it, where we combine the two proteins, we call it an interface. So the interface, it is the part of this complex, protein complex, that is buried or within that is taking part within the reactions of between producing the interface, protein-protein interface between the two polypeptide chains. As you can see here from the diagram, we have two polypeptide chains where some of the residues from the two polypeptide chains get into non-covalent reactions, and this, where they combine together, we call this part is the buried part because it's not exposed to the solvents within the cell. So this part, the buried part, we call it an interface. Okay, so buried surface or an interface, it's the same name that is termed interchangeably between that designates the reaction between two polypeptide chains due to the reaction of the side chains of the two polypeptide chains with each other, and they are producing what's called the interface area. And this is a buried area that is not exposed to the solvents of that is where it's found, these proteins within the cell. So the interface are usually composed of hydrophobic amino acids like tyrosine, histidine, tryptophan, or phenylalanine. And also there are charged residues. They are less abundant except for arginine. It's also found within these interfaces. So this, if you notice that the tyrosine, histidine, tryptophan, and phenylalanine, these are hydrophobic amino acids. That's why they tend to be away from solvents. So that's why they get into reactions to produce what is called an interface. So the production of the interface, or once that these polypeptide chains get into an interaction, their geometrical and structural and conformational change because of this interaction between the two polypeptides. So when you study, you need to study the protein geometric, the complex geometrical and structural and dynamical aspects, so that you can note the function of this protein complex of the two polypeptide chains once they get into an interaction and produce what's called an interface area. So what are the factors of protein-protein interaction interface? When we are defining an interface, we need to define the surface area that is buried by by the interaction between these amino acids. And when we define this area, we need to define what is the fraction of non-polar residues that is part of this interface. So usually this interface consists of hydrogen bonds and salt bridges. So the composition of the interface depends on the type of bonding between the two polypeptide chains that lead to the buried surface of this interface that is found as part of the surface area where the interaction takes place. So, as I mentioned, the binding affinity between the two polypeptide chains defines the strengths of this interaction or this interface, that depends on the type of amino acids that's involved within this interaction. Okay, once this interaction and the interface production, this results into different conformation of this polypeptide chain or this interface. So this defines the secondary structure of the original secondary structure of the polypeptide chains, if they have beta helix or beta sheets or alpha helix. And this again defines the protein stability of this interaction, depending on the type of the amino acids that's found within the interface. So the geometric and electric complementarity is very important between these two polypeptides that define the strengths of this interface. And we call this strength as binding affinity between the two polypeptide chains. Within the interface, there are different types of atoms. As you can see here from the diagram, these are two polypeptide chains. This is the interface between the two polypeptide chains, and we call this an protein-protein interaction, because there is a production of an interface between the two polypeptide chains. So from this interface, we define what we call the interface atoms. So the interface atoms, these are all those atoms that lose solvent accessibility because they are buried within the interface. They are in reaction with the other polypeptide chain, so they are not exposed to the solvents within the cell. So this type of buried atoms, like A, B, and C, all these are called interface atoms. Some of these atoms, they are fully buried, and others are partially buried. That means If they are fully buried, they are not exposed at all to the solvents. But if they are partially buried, then they are partially accessible to the solvent of the solvent in the cell. So those are fully buried, like B. These are fully buried because they are not exposed to the solvent at all. But like C and like C, it is partially buried. So these are all contact atoms. So usually the C, it's a type of amino acid that undergoes like van der Waals reactions with the other polypeptide chain. So they tend to be at the rim or the periphery of the uh, of the of the interface. And this type is usually weak bond type of weak bondings. So they are exposed to the solvent, and they sometimes lose their bonding with other polypeptide chain. But at the end, we call them within the interface, as part of the interface, but they are not fully buried, they are partially buried. So before we talk about hotspots, do you have any question up to this point? So before we move on to the rest, do you have any question? Okay,
No question, Prof.
okay, thank you. So we move on to define what is hotspots. So hotspots are the regions on the protein-protein interface responsible for binding. As I mentioned before, the interface, protein interface, is the area where the binding, where the two polypeptides contact, let's say, contact each other. Okay? But some of the amino acid residues within this interface, they are not part of this bonding. They are found in the vicinity of this interface, but they are not taking part in this bonding. So this type of residues is not called the hotspots. Those which are taking the binding, we call them the hotspots. So hotspots are the region on the protein-protein interface responsible for binding. Not all residues that are present at the protein-protein interface contribute equally to the binding free energy. And we defined before what is binding free energy. That is the difference of energy production between being the residue being unbound and the binding status of these residues. So there is a production of energy, and even when it gets into binding, there is use of this energy. So the difference between being unbound and bound, this is what we call the binding free energy. So to study this, or to note what are those residues that is taking part in the binding, there is a process what's called a procedure that's called alanine scanning. They use what's called alanine scanning. So as you know, alanine, it's an amino acid. It's a non-essential amino acid, but it's called an inert amino acid. That means it doesn't go into any reactions. It has a methyl group at its R chain side group. So, but once it is introduced within the polypeptide chain, uh, we call it an inert amino acid. It doesn't change the conformation of it neither change the conformation of the polypeptide chain, nor it gets into reactions with other amino acids side chains. So its presence actually, um, it doesn't have any effect, but at the same time we use it here to replace certain amino acids within the polypeptide chain to check if this polypeptide chain, it goes into reaction once it is substituted by this alanine, the certain amino acid substituted with this alanine, or it will remain as it is. And the difference between the wild and the mutated one that is introduced alanine, is it shows which amino acids that is responsible for the binding of or producing the interface between the two polypeptides. That means we are mutating certain amino acids within the protein, the polypeptide chain that we need to study, by introducing alanine instead of the active amino acid that is found in the polypeptide. They were replacing with alanine, and checking if this polypeptide chain goes into interaction with the other polypeptide chains. If it didn't go, then this amino acid that is replaced is the responsible for binding. If it goes into interaction after the mutation or the replacement with alanine, that means this amino acid that is replaced, it is not responsible for binding interaction, because it's still without its presence, it goes into an interaction. And this process, we call it an alanine scanning. So those small residues at the protein interface that contribute to the binding, we call them hot spots. So the interface, as you can see, it consists of a rim and a core. A core is the central part, and the rim are the amino acids that's within the interface, but at the periphery of the interface. So the core is always rich of hydrophobic residues, and as I mentioned before, they are the buried part of the interface. And amino acids compositions of the rim and the rest of the amino acids which are in the rim is the same as the rest of the protein. That means they are partially exposed to the solvent. So hotspots, from different studies, they found that hotspots, or the residues that goes into the binding or produce the binding effect within the interface, tryptophan is the highest amino acid that is leading to this binding. That means it has high binding affinity in producing the interaction between two polypeptide chains. And then comes the arginine and tyrosine residues. So these three amino acids are very important in binding between two or maybe more polypeptide chains. So they are the one that defined as hot spots. There are less other amino acids, but they are less frequent, like methionine, serine, threonine, and valine, are also found in hotspots, but less frequent. So the hotspots regions are more tightly packed as compared to the rest of the interface. So as I mentioned, these are the type hotspots if in very simple words, these are the type of amino acids that that have high binding affinity, leading to tightly packed interface between two polypeptides or more than two, and these are specific types of amino acids with high frequency found within the interface, which are tryptophan, arginine, and tyrosine. Okay, mechanisms of interactions, as you can see here from the different types of diagrams or represented diagrams, we have, if the type of amino acids are non-polar residues, this can produce what we call hydrophobic interactions. And this, of course, they are buried, and they are the solvent is it's hydrophobic. That means they are repelling water, and so these are buried within the interface, and they are not and they are not accessible to the solvent. If those taking part in the interaction are polar residues, then this facilitates either electrostatic interaction like van der Waals interactions or hydrogen bonding. So it depends again on the side chain of the amino acid, which is a polar residue, but it depends on the side chain. In the other types, those that utilize metals like zinc, magnesium, the presence of these metals is important to facilitate the interaction between two polypeptide chains. And this is, we call metal cofactor sharing. So it facilitates the interaction between the two polypeptide chains or the side chains of the amino acids of the two polypeptide chains. The type of covalent bonding, as I mentioned, it's not highly frequent, but it is evident within the protein-protein interactions, is the type of disulfide bonding or bridges, and this leads, of course, to oligomerization between the two polypeptide chains, and it's a strong type of or permanent type of protein-protein interactions. Other type of interactions, it's the architecture or the conformation of the protein. So the structure of the grooves and the three-dimensional structure of the proteins itself can be part of can lead to the production of protein-protein interaction. So as you can see here, if like B is fitting within the architecture of the conformation structure of the other polypeptide A, and so the conformation structure itself can help or can facilitate the interaction between proteins. Like, for example, in B, because there is a difference in the conformational, so they cannot fit together. So this type it depends between a chemical interaction rather than physical interaction or fitting together because of the architecture of the protein or conformation or three-dimensional structure of the polypeptides. Okay, the protein-protein interactions, it has different types and are grouped according to they are grouped according or based on the certain characteristics. So first, based on the chains or the polypeptide chains identity, if the polypeptide chains are interacting with each other are similar, then we call them homo-oligomeric. If they are different, then we call them hetero-oligomeric. The homo-oligomeric, they are either isologous or heterologous, and this is based on structural symmetry. Okay, we are going in the coming slides. We are going to define this in more depth. Heterogrammic, it's non-obligate or obligate based on chain affinity. Okay, so that's mean the binding affinity. If they are not non-obligate, okay, either they are permanent or transient, and this is based on their stability, how much they are stable together. If they are transient, then based on the interaction strengths, it could be weak or it could be strong or weak. So this is the different types of interactions, protein-protein interaction, based on different characteristics, either on the chain's identity, that means the type of amino acid residues, identity between the two proteins interacting with each other, or based on the symmetry, structural symmetry, or based on chain affinity, or the binding affinity as we call it, or based on the complex stability after they combine with each other. Are they stable? And this take parts, different factors take part within stability, defining the stability. Or they are, the interaction itself, is it strong or weak? And each one has its own definition. So when we say homodimers or homo-oligomeric or hetero-oligomeric, it's we are talking about the resemblance of the protein cores between of the two interacting proteins. So if the area of the interaction, it's more similar, typically large hydrophobic amino acids, then leading to non-polar buried surface areas. So this shows good complementarity between the two chains. If they are heterodimers or hetero-oligomeric interfaces, then these have large hydrophobic and charged polar residues. And these residues are more frequent exposed and non-interface surface, because they are not complementing each other. So that means there is a difference between the two polypeptide chains. Obligate and non-obligate. So we define the interface, or sorry, the interaction as obligate. That means cover complexes of promoters. So when you say promoters, that means protein complexes. It's the same meaning, that cannot exist independently. That means they have to be combined with each other to function as a complex. But if these complexes, each polypeptide can exist independently, then we call this non-obligate complexes. Another distinction is permanent complexes, and these are strong and irreversible. And usually this type of bonding that is part of this interaction, usually this is a covalent bonding. Transient complexes, in the other definition, opposite definition, these are complexes that are readily changeable. That is, they can change in in a continuous manner, and this is why they are called transient. For specific time, then they change their status to a different status, and they might return back to independent status. Uh, Even if they are transient, they could be weak and they could be strong. So weak, this is defines the dynamic or the dynamic of reaction between the two or the three polypeptide chains or more. Maybe the depends on the number of polypeptide chains that's part of the interaction. So this is again defines the dissociation constant. So in the weak, the lifetime of this dissociation is in seconds. In the strong, it's more. It might take more longer time, and we define this. It is strong, but yet it is transient complexes. So this defines weak and strong defines the dynamic of the interaction. As you can see here from the diagram, the binding affinity from permanent to weak transient drops. So more if there is a more binding affinity between the residues of the interacting proteins, then it goes to the association between the two proteins or more proteins tends to be permanent. But when the binding affinity decreases, then it becomes weak and transient. So in this slide, we just define as the what's homodimers or homoligmeric and heteroligmeric. As you can see here from the diagram, that if the proteins, interacting proteins, they have the same sequence of amino acids or they are similar, then we call them homoligmeric. If they are different types of proteins with different amino acids, then we call them heteroligmeric. So this again defines the type of interaction between the proteins, because there will be different types of side chains if it is heteroligmeric or homoligmeric. Structural symmetry. Again, this is another character or defining character of the interaction, protein interaction. And as we mentioned, if they are homoligmeric or homologous or heterologous, then the symmetry, structural symmetry, define if it is isologous or heterologous. And this is very important because it shows the aggregation between the polypeptide chains, if it is the symmetry between the polypeptide chains, and this is very important in the defining the conformation and structure of the proteins. So isologous, they are symmetrical in very simple words. Heterologous, they are non-symmetrical, and we define symmetrical and non-symmetrical by a perpendicular plane of the protein final conformation and structure. Chains affinity, it's either obligate or non-obligate. So from the diagram here, you can see the obligate, they are interacting and they are, there is a strong association between them. and and again defines this association is that they have longer lifetime and they are more stable. But while the non-obligate are usually not less stable, and they tend to dissociate in a very dynamic character, because they don't have high affinity to each other as obligate. Again, the stability defines the association as permanent or transient. So permanent interactions are very firm, exist usually in the form of complexes, and this is like these ones. They tend to interact with each other in strong bonding types. Who are transient, they have the tendency to dissociate and reassociate again in vivo. So they are not very much, they don't have very high binding affinity. The other type of character of the protein-protein interactions is being weak and strong. They are transient, but they could be weak or strong. So in weak transient interactions, as you can see here, the example, the oval albumin and the lysozyme. These are different proteins. They interact with each other to give over-lysine complex. But this over-lysine complex, its stability is defined by the pH of the cell or the solvents within the cell. So in acidic pH, they are stable, okay, and as the pH goes to higher, to alkaline pH for 12, then they tend to dissociate. So again, this is called transient because it changes with the pH of the cell or the solvent where it is found, this complex. So its dissociation depends on the pH, and as you increase the pH, then to an alkaline status, then they tend to dissociate. So in weak transient interactions, there is continuous formation and breakdown of interactions. While in strong ones, the interactions require a greater amount of molecular provocation to break. Okay, we define the different types of protein in action. So then you, after defining these types, then you can study the protein interactions. You cannot study protein interactions and you don't know about your protein, it's the type of interaction it undergoes, the chemical nature of these interactions, the physical and chemical properties of these interactions. So you need to define first what protein you are studying, and its association with other proteins or interaction with other proteins. This is very important, so that this will help you to select the method you need to study this protein. So studying protein interactions, why do we study this? Because protein interactions are essential for protein function and cellular pathway formation. This is very important. Since these interactions are also involved in the development of diseases, okay, they are important targets for drug design. and artificial design of protein complexes. As I mentioned before, it's not only for human, it's not only for animal, even for the environment, for plants. The study of protein action is very important. Otherwise, if you study the protein as an isolated component, then you will be faced with the reactions of this protein with other proteins once it is introduced in vivo, and could lead to different side effects and adverse side effects that you didn't count for while you are studying the protein as a separate entity. So for studying methods, you need to assess first. Usually, the methods assesses if this interaction really exists or not. That means you first need to know if this protein goes into interactions or not, and this depends on the studying. Is depends on the sequence, the primary structure of the protein, its secondary structure, and its and if it has three conformation structure and its side chains of these amino acids. So if there is an interaction or not, if it is evident or not, and this usually is studied in vivo. And the second assessment, where does this interaction takes place? Is it in the nucleus? Is it in the cytoplasm? Is it in the plasma reticulum? Or maybe it's in a prokaryotic cell? And you need to know if it's in the chromosome. If where does it take, and with which protein? The other question you need to ask is how does it take place, this interaction? Is it through covalent bondings? If it's non-covalent, and what type of non-covalent? So this is one of the questions you need to ask so that you can select your method. And how much the dynamic of this interaction. So the binding stability of the protein complex. Is it a permanent? Is it transient? Because again, selecting the method of studying a protein interaction, it depends on this, asking these questions. If it's evident, where, how, and how much the dynamic or the stability of this protein interaction. There are different methods for detecting protein interactions, but mainly grouped into two categories: experimental and computational. That's why we define these methods are defined as in vivo, in vitro, and in silico. And can anyone define for me what's the difference between in vivo, in vitro, and in silico? Can any one of you? I need to hear your voice. What's in vivo, what's in vitro, and what's in silico?
I think when we are talking about in silico, we are talking about using the computer.
Good.
While when we are talking about in vivo, it's when you're doing something in a live animal.
Yes.
Or in a plant, I think. When we're talking in vitro, you're talking about in a test tube, when you're doing things in the lab.
Great. Great answers. So in vivo, within the cell, as your colleague mentioned, or within a live animal or a plant, a live cell, okay? And even if you are using a tissue culture, once you are using a cell, this is considered in vivo, even if you are doing this within a lab. So as long as you are using a live cell, it's a mammalian cell or a whole animal or a plant, or so these are all in vivo. But once you, for example, you extract a protein and you are, for example, quantifying a protein in a lab, or studying a protein using electrophoresis, for example. These are all, we call them experimental work. It's in vitro. And once you use computer programs or servers or databases for analyzing, for example, a sequence of amino acid or a sequence of nucleotides, DNA sequence, RNA sequence. So you are studying in silico. So this is very important. You need to know the difference between this, because this is the first step. You need to define what medium you want to use. Is it in vivo, in vitro, or in silico? Okay, so for protein interactions, so we have experimental, and this again, experimental could be in vivo or in vitro, and computational, as it's in silico, and this is using different databases or servers or programs or softwares to predict the because these are all using algorithmic calculations and modalities and models to predict structure of a protein, to do docking, and to predict the protein interaction with other proteins and so on. But it's again, it's a valid studies that help the researcher to understand more the proteins once they have their sequence available. So these tables, I know it's small in size. I couldn't make it more bigger, but once you print it, you will be able to study it. So these two tables shows compare the different protein interaction methods, and the blue writing. I added this blue writing to give more insight about these tables and the methods, the different methods, because some are in vitro and some are in vivo and others are in silico. So for the first, and you need to know this table very well, because this is the base of the different methods. Actually, this is the summary. That's what you need to know about the different methods. So, for example, for the first one, the fluorescence resonance energy transfer method. For example, the principle behind this method is non-radioactive energy transfer between a donor acceptor fluorophore when they are in close proximity. So in this way, to study the protein interaction, you use a fluorescent type of chemical that gives light. And if the interaction happens between the two proteins you are studying, of course, it has a workflow. We are not going to talk about the workflow, but we are going to select some of these methods and have more insight in it. But you need to know the principle of each method, and as it is defined here in the table, the principle, the advantage, disadvantage, and applications. So the fluorescence resonance energy, we use it for intercellular protein interaction to detect the intercellular protein interaction, especially those lead to the pathway of signal transduction. And it is widely used in signal transduction research. So this method it involves both in vivo and in vitro. We can use the cells, or we can use extract the proteins and study it as separate proteins and their interaction within the lab as solutions. And then we can process it with different methods, electrophoresis or chromatography and so on. So to this step, you don't need to know the workflow, but you need to know the principle and its application, and its advantage and disadvantage. And the most important, is it a in vivo process or in vitro process, or is it in silico process? The other method, for example, is the biomolecular fluorescence complementation, and this is it uses again fluorescent dye or protein. These are tagged proteins with fluorescent dyes, and then they are studied to see if there is a complementarity between the protein tagged and another protein, to detect identify, for example, a novel interaction. You don't know if there is an interaction or not, but you are testing a protein against other proteins. And this type it will be screening the different genes, the production of the outcome of these genes, their transcription and their translation, and then you are testing against a known protein. So you want to test if there is an interaction or not. So here, as if you are asking a question, if there is an interaction or not, you don't know. So you are detecting a novel protein-protein interaction. And this is usually done in vivo, because not only the interaction can take place with respect to other factors that's available within the cell. Such type of experiments, you cannot do it in vitro, because you need to see the interaction as it happens within the cell. That's why this type of experiment, for example, it's in vivo, and usually it is testing for transient protein-protein interaction, because you are testing a protein against other proteins, and how, as if you are testing a protein, how it behaves with other proteins. So studying a protein and its behavior with other proteins, you tag the protein with fluorescent dye, and once the reaction, it interacts with other protein, the dye gives the light on fluorescence and can be detected with fluorescent microscope to see if it is action. And then you can proceed to understand more what type of this interaction it is with other techniques. Proximity labeling, again, this is another type of method, and this is again in vivo and for transient protein-protein interactions, and this all actually tests the dynamic. So as if you are asking the other question, how much is this interaction? So it tests the dynamic. Again, it depends on labeling a protein using an enzyme, okay, and then testing the type of bonding between the proteins and the tag protein, and this gives more information about the dynamic of the interaction between the different proteins. As you can see, these methods differ its principle from each other, depending on what type of question you're asking or what's your research question. Are you looking for studying if this interaction exists or not, or the dynamic of the interaction, or the type of interaction, and so on. Again, there is another method in vivo, and this tests both transient and permanent. It's called the EASE-2 hybrid method. And again, this is using the transcription processes within the E cell. And again, we call it in vivo because we are using the E cells. And this tests protein interaction network mapping. That means a protein, once it is tagged by a certain DNA binding protein, then we test if it helps in the transcription. Once it is transcribed, then definitely it is translated. So this translated protein, we are testing how it is produced. Is it a combination of proteins that give rise to what we call the transcriptome in the production, or so we are testing the network mapping, which is testing the gene function based on the transcriptome production. So and this is why we use the yeast, because the yeast, it has different types of genes that needs a combination of proteins to combine together to activate a gene to be transcribed and then translated. The other types also that are in vitro. In vitro, for example, the GST pull down assay. So this method, it's in vitro because we are extracting a protein, and this protein is tagged by GST, which is guanosine transferase, and is mobilized on a medium that contains different beads, and then different type of beads that is adhering the protein to a surface, and then we incubate it with lysates to capture and interact with proteins. As if you are attaching the extracted protein to a surface, just like chromatography, to a stationary surface, and let the other proteins interact with this protein and test if the type of interaction. So you use this method in vitro to verify a non-protein against non-known or unknown proteins. So that's why it is in vitro and testing this interaction. And then you can, after you study it in the vivo, then you can test it in the in vitro. So other types of methods like the mass spectrometry, it's in vitro. This is mainly used to identify the type of proteins based on the structure of the amino acids, the ions of the amino acids. So you are actually mapping a protein within a large scale or a complex environment. So this always used for defining networks, mapping networks. So a protein, it's mapping with, or type of interaction with other proteins within a complex environment within a cell. We are going to have more information about mass spectrometry when we speak about the proteomic methods, so you can understand more about mass spectrometry. The last method is a computational prediction, and this depends on bioinformatics. Definitely, we'll have or I don't know if you already have it or not, about bioinformatic tools and machine learning tools. All this help in predicting a polypeptide chain from its amino acid sequence, predicting its structure, its interaction with other proteins that can be extracted from the databases, and even the defining the three-dimensional of this structure of this complex, and you can move into docking and designing drugs or protein complexes for therapy, or as drug for diseases, or supplementary proteins. So one of the in vivo methods we are going to talk more about. We are going to select 2 in vivo methods and 2 in vitro, and give more insight on the computational methods. But before we go into the in vivo methods, there is a principle for these in vivo methods. All methods are based on a reporter system. Okay, all the in vivo methods to study the protein-protein interaction, they are based on what we call a reporter system. This reporter system consists of 2 parts. As you can see here from the diagram, one part is linked to one of the proteins of interest. Okay, and this is called bait protein. Okay, and the other one is linked to another protein of interest. These are the proteins that you need to study. Okay, so one is the bait. The bait is the protein, one of the proteins you want to study, and the other protein, the prey protein, it is another protein. You want to test the interaction between bait and prey, but you link them to a protein within the cell that already interact. So when both proteins interact with each other, the 2 parts of the reporter system are brought into proximity and have a clearly detectable effect. So the importance of this reporter system is that you select 2 proteins within the cell that you know they already interact. The benefit of this interaction of these proteins, you want to make use of this interaction to get the 2 tested proteins together in proximity to each other. If they interact, then they produce a signal. If they didn't interact, because here you are sure that they are proximal to each other. If they didn't interact, that means there is no complementarity between them. There is no binding affinity between them. So they don't act. So the term bait and prey are used is a protein of interest, and the objective is to hunt for interacting partners, as if you are making them come together and test. If they interact, they have a binding affinity, they will interact and produce a signal. If they didn't interact, then they don't have binding affinity, and there is no signal will be produced. Okay, then for example, one of these examples that use this reporter system is the Yeast-2 hybrid. So the principle of this system is that, or this method, relied on the fact that the DNA binding domain, okay, the DNA binding domain and the transcriptional activation domain, okay, of Saccharomyces cerevisiae, the yeast, transcription factor GLA, okay. So the GLA transcription factor, it will not be activated unless there is a combination of proteins that come together, like this one. This is a type of interaction to activate the enzyme polymerase, DNA polymerase 2 enzyme to start the process of transcription. So this type of interaction, it's important for activation of the polymerase. Like here in A, there is no interaction between X protein X, Y, and AD, which this constitutes the transcriptome for the reporter gene to be transcribed. Because the interaction, it has to take place to activate the DNA polymerase enzyme, the RNA polymerase enzyme to transcribe the reporter gene. So in turn, the reporter gene activity allows one direction interaction between the proteins. So X, Z, and AD, this type of interaction between three proteins, and the same time their importance for activation of the transcription enzyme to start the transcription of the reporter gene. So this is important. This is the base of the principle of these two hybrid. But I want to show you a video here. Okay, let's see it.
And now,
Okay,
back
sorry
to
for
Saved by
this.
Kinsta.
For this,
Dinner time, Zach.
Many a times in a cell, proteins need to interact with one another to bring about a specific
can you hear, or is it clear? Can you hear the voice?
Yes, we can.
function like activation or inactivation of target proteins. Yeast-2 hybridization is a molecular biology technique to validate protein-protein interaction through activation of a reporter gene. Y2H is extremely sensitive, cost-effective, and very easy technique to study the protein-protein interaction. This technique is based on the reconstitution of transcription factor fragments which are fused with two different proteins whose interaction needs to be studied. In this figure, a gene and its upstream activation sequence is shown. A transcription factor comprises of DNA binding domain and activation domain. The DNA binding domain binds to the upstream activation sequence, and now the activation domain recruits the transcription activation machinery to activate the transcription of the gene to produce mRNA that later translates to protein. In yeast-2 hybridization, the two domains of transcription factor are separately cloned in different plasmids. The first plasmid has binding domain fused with the target protein sequence termed as BAIT. The vector also has tryptophan gene to select the transformant on the tryptophan lacking media. The second plasmid has the activation domain fused to the second target protein sequence termed as prey. In this, leucine gene is used for transformant screening. Thus, plasmid 1 produces BD BAIT fusion and plasmid 2 produces AD prey fusion. A genetically modified strain of yeast deficient in tryptophan and leucine is used to select successful transformants on selective media lacking tryptophan and leucine, without which the yeast cells cannot grow. Now both the plasmid and the yeast are inoculated in the selective media and later plated on the solid agar plate containing leucine and tryptophan deficient media. After incubation, only transformed yeast cell containing both the plasmids will grow into the larger colonies. Now what happens inside the transformed cells? The product of the first plasmid, that is fused BD and BAIT, comes and binds to the upstream activation sequence. Now if the second target protein, prey, interacts with the first protein, then the BD and AD fragments come in close proximity, leading to the recruitment of transcription activation complex, which brings upon expression of the reporter gene. And if both the proteins, that is bait and prey, do not interact, the two domains of the transcription factor, that is BD and AD, never come in close proximity, and as a result, the expression of reporter gene is not seen. Expression of reporter genes like the GFP, GUS, or LEGZ are observed under fluorescence microscope or visually. If you want to know more about GFP, watch.
Okay, I think it's very clear now. And as you can see, it's, sorry. As you can see, the yeast is used as a medium for testing the interaction between two proteins. But we are using what the principle behind this, we are using the already available pathway of transcriptome production. So we know that in the cell that there is a transcriptome proteins, they come together, they interact together to activate the transcription of a reporter gene. So we are making use of this sequence of these transcriptome proteins using the plasmids to express themselves, but they carry the different proteins, they carry with them the bait and the prey proteins that we are testing if they come into interaction or not. So this is the base of the principle behind the two hybrid. So, so they are using a reporter system. If they come together with the presence of the bait and the prey, they are interacting, then there is a production of the reporter gene. There is the production of the reporter protein, which is transcribed because there is an interaction between the proteins. If there is no interaction between this protein, then this stops the interaction and stops the transcription. So this is mainly the base behind the yeast to hybrid principle. The other method, which is in vivo, the co-immunoprecipitation method. So this is widely used in vivo technique for detecting physical protein interaction. Again, the method relies on the reporter system, and it has different steps. For example, here in the plant, they are using the plant tobacco. They mix filtration solutions. They use the agrobacterium species of bacteria. They take the tobacco and they prepare an extract, and they inoculate this with the bacterium, and then they extract this bacterium. It utilizes the tobacco, and then they prepare a protein crude out of the extracts, and it is labeled with different labels, probes. We call them probes, and then using the agarose we separate. If there is the probes, if there is an interaction between the different proteins, and the tobacco is just to stimulate the interaction between the proteins. So so it's if there is an interaction between the two proteins you are studying, then we use the agarose or the electrophoresis to separate the proteins. The molecular weight of if there is an interaction and production of a protein complex, the molecular weight will differ, first of all. The other thing is there is no interaction, then you will find two separate proteins. Then you can verify by Western blotting. We are going to talk about Western blotting using a specific antibody that interacts with these proteins to validate if it's a separate protein, independent, or it is the complex. So it's like an antigen antibody reaction, but using a protein that is separated by electrophoresis. So this reflects protein-protein interaction occurring in living cells or organisms. However, co-precipitation is unable to determine whether bait and prey association represents a direct physical contact or requires an intermediary protein for complex formation. So it shows there is an interaction, but there could be an intermediary cofactor, like a metal, like another protein. But if there is a what we call binding affinity between the proteins, then the molecular, once you separate it by electrophoresis, the molecular weight will differ from the separated or independent proteins, each one separately. Definitely their molecular weight will be smaller, but once they combine, their molecular weight will be larger. To validate that this large molecule is from the combination or the interaction between the proteins, you need to do Western blotting. So Western blotting is for validation. And as I mentioned, this is one of the proteomic methods. We are going to speak about it more when we come to the proteomic methods. We will talk more about Western blotting, which is an antigen antibody verification method. One of the in vitro methods, what we call the GST or glutathione S transferase, and this is an in vitro technique for studying protein-protein interaction. It uses a reporter gene, a reporter system, and the bait, which is one of the proteins that you are studying, is often tagged with glutathione S transferase or histidine or other affinity tags. And this is immobilized on beads coated with specific ligand, and then incubated with the prey, and then testing the binding partners as are captured through direct physical interaction. So after washing, if there is a binding, you can take after you wash, you take the eluted proteins and separate it by SDS page, which is sodium diethyl polyacrylamide gel electrophoresis, or mass spectrometry. So this is totally in vitro. It doesn't use any cell or live cell. So you are testing the interaction in the lab using normal chemicals and plates. Of course, it has a lot of measures, testing the different pH, testing the concentration of the solutions. It's a tedious process, but yet it's one of the processes that is studying protein-protein interaction is in the laboratory without the use of live cells. So in this case, you are testing a known protein against an unknown proteins. So it's a type of demonstrating possible interaction between two known proteins or unknown and known protein. Mass spectrometry, it's more like analytical rather than testing an interaction. So after you proceed with your different methods of testing the interaction, if it is answering the first question, if there is an interaction or not, then you can detect you can study more about this complex using mass spectrometry. So you need to enrich first your proteins and test their interaction, enrich proteins with their binding partners. That means if you know that this protein is binding with or interacting with a certain protein, you enrich this within the laboratory using the different methods like the pull down assay just now we mentioned, or affinity process that is interacting the two proteins and enrich them. And then this complex of protein, you can study it more with mass spectrometry. So the use of mass spectrometry, it identifies the type of interaction between the two proteins. If it is covalent, non-covalent, it needs a mediate or not, and it identifies the regions, protein regions that, or the residues that, or defines the hotspot as we call it before, and the type of these residues, the different type of post-translation modification that is taking place within these residues that help in the interaction. So it gives you more information about this binding interface, the type of amino acids, the type of interaction, the chemical bonds, the physical structure, the ions, the modifications. So it gives you more studies about the type of interaction. The in silico methods, it's a computational prediction, and this, of course, it depends mainly on predicting protein interactions, predicting using bioinformatics and machine learning, which is the AI use. So what can you predict? You can predict from the sequence, the structure, the evolutionary relationship between these proteins that's interacting. It will give you more information about the function, what could be the function according to the domains found within these proteins and the type of interaction. It gives you more about the network properties. If this protein interacts with other proteins and form a network. So these computational methods or tools, this is provided through servers, and there are different softwares and servers. Some of them, they predict protein structure interaction. Others, they give functional annotation and classify these proteins or this type of protein interaction, if it is permanent, if it is transient. It gives the dynamic behavior. It simulates the dynamic behavior. You can see the interaction with its charges, its interaction with the solvents, how it changes with pH and so on. It gives you full simulation about the type of interaction and the factors that affect for this interaction between the proteins. It gives you more other servers. It gives you functional network and pathway analysis. So pathway analysis identified by linking this interaction with its biological function. So the application of this computational prediction, it's important. Now it's to predict the drug targets and binding sites. It evaluates the structural and functional effects of disease mutations, and it designs artificial proteins or optimize enzyme activity. So this all with prediction, but they are very helpful and valuable. And from these studies, you can start with these studies, and then you can go further into the in vivo and vitro to see these reactions and its effect on in a biological setup or life setup. It's the same. This diagram shows the different input you need to provide to the software or the service, either the sequence or gene ontology or protein structure. And then you can start with any input of these, but again, it will give you, according to their input, it gives you different prediction information as you according to the server and your research question. But what is important here is the network-based features. This use different coefficients, different algorithmic functions, and there is a difference between these algorithmic functions, like the Jaccard coefficient is a metric used in protein-protein interactions network analysis to quantify the similarity between two sets of proteins. So it gives you the similarity index. So measuring the overlap of their shared interaction partners. ADMIC, other index, is a topological similarity metric. So each one, it has its own use or its own output used in link prediction of the protein-protein action networks to predict unknown or missing interactions. So it identifies potential protein pairs by calculating the similarity between them based on shared neighbors in the network. So it gives you it extracts from the database and shows the network with a certain protein, their input, and gives you more information about the type of network. The common neighbors, is a key topological feature used for link prediction and identifying protein complexes based on the assumption that proteins sharing many common partners are likely to interact. So this is very important for predicting the network-based functions of a protein. When we are talking about multiomics, usually the combination of methods is important. For example, you can predict, but you need to combine it with other in vivo or in vitro methods. So single approaches can be biased and limited, but when taken together, they can provide a more comprehensive view of biological networks. As I mentioned, the prediction itself is not enough. It's useful, but it's not enough. You have to undertake in vivo because testing the protein in its normal environment is important because there are other cofactors, as we mentioned before, affect these protein-protein interactions. So you need to have full study, full overview of your methods and its protein and your protein under study. So this diagram shows other methods and how they interact. For example, if you are working on weak transient protein-protein interactions and you want to see their functional association, you need to study beside the prediction, you need to study the functional association within the in vivo. So, or do evolutionary studies with other proteins, how this protein evolved. And so it's important to link your study with other proteins, with other methods. So here is the assignment for this week. I will send this today for you, at least the presentation, the PowerPoint presentation, and the record get from the Google Meet. But this is a question for you assignment. You can send it to me by email. Define in one paragraph, maximum 200 words. What is meant by protein-protein interactions, main interaction points. I mean here about the hotspots, main function of complexes, and the basis of classification of protein-protein interaction complexes. Any question up to this point? Do you have any question?
No question, Prof.
Okay. I'm just going to take one of the methods of proteomic methods. There are a lot, but let's start for this session and then we complete it in the next session. For the proteomic methods, there are different methods according to the purpose of the objective behind using this method. What is your purpose or the research, your research question? Are you looking for purification or for analysis of a protein or characterization of a protein or structural analysis of protein or quantification of the protein, or you want to study the sequence of a protein? So based on all this, you can end up with the protein sequence, with the structural analysis, and then you can use all this data to and then you can use all this data to use bioinformatic tools for further analysis or prediction. So I'm going to start with the chromatography for today. Okay, before I start with the chromatography, as you know, to study the type of amino acids within a protein, you need to you can use a biochemical test in the laboratory, which is a hydrolysis test by heating the protein under study with 6 molar of hydrochloride acid or hydrochloric acid for 14 hours at 110 degrees centigrade. So usually the amino acids will hydrolyze, and then you can separate them by chromatography. Okay, you can use HPLC, which is high performance or high pressure liquid chromatography, and this we are going to speak about it. So the amino acids, knowing the type of amino acids, it's a first step for studying a protein. You need to know what type of amino acids. Then you can study the sequence of these amino acids. This is another step. And as we mentioned before, we explain the Edman process for knowing the sequence of the amino acid using the enzymes trypsin and digesting the protein into small peptide chains. So this is a very quick process within the lab. It doesn't take long. As the chromatography HPLC itself, it's a quick process, but your solution, it has to be clear and clean for the process to run, for the HPLC to give you good results. When we speak about purification, we speak about chromatographic techniques. So the chromatography, it's one of the methods used in the laboratory and mainly depends on, as you know, on 2 phases, a mobile phase and a stationary phase. So all chromatographic system consists of the stationary phase, which may be a solid, gel, liquid, or solid liquid mixture that is immobilized, and the mobile phase, which may be liquid or gaseous, and which is passed over through the stationary phase after the mixture of analytes to be separated. So the separation of these proteins within a mixture of proteins depends on their solubility within the mobile phase. So the more they're soluble, they move. The more they are less soluble, they tend to separate into different zones, depending on their solubility coefficient to the mobile phase, which could be a liquid or gaseous. So during the chromatographic separation, the two phases, the movement of the mobile phase distribute the proteins according to what we call the distribution coefficients. And this distribution coefficient again depends on the solubility or their interaction or their chemical reaction or their movement with the mobile phase. And this tend to separate them according to their again to their chemical properties of the proteins, that is their chemical and the physical properties of the proteins in the mixture of under study. Here, this table summarizes the different methods of chromatography, and I believe this is a very good table. It helps the researcher to select which method I can use, because there are different types of chromatography. There is the thin layer chromatography, there is a paper chromatography. It depends on the stationary and the mobile phase of chromatography. So these different types of chromatography it depends. Your selection depends on the protein, the physical and chemical, the physical and chemical properties of your protein under study. So accordingly, you can select the stationary phase and the mobile phase, and this defines the type of chromatography. So, for example, here the thin layer chromatography, paper chromatography, and reverse thin layer chromatography, their uses most frequently used on a small scale to identify unknown compounds based on the retardation factor. That means the retardation factor, it helps. Retardation factor, that means how their reaction with the mobile phase. So this is the use defines the method you want to select. The commonly used in the labs is the high performance liquid chromatography, or sometimes they call it high pressure liquid chromatography. Is the most popular and efficient way of chromatography. Is an analytical technique used to separate, identify, or quantify each component in a mixture. So it separates, it identifies, and quantifies. So three outputs out of this method, because this is linked to a computer that quantify, it separates, it identifies because it has a database, and it's the system itself is separates. It's based on the separation, its movement in the mobile phase. So let me show you a video here for quick.
And now, back to Saved by Kinsta.
Let's
Everyone
show you.
under
Hey everyone, quick biochemistry basics here. Let's talk about HPLC. HPLC is also known as high performance liquid chromatography or high pressure liquid chromatography. HPLC is usually a modified column chromatography. In column chromatography, a column is packed with an absorbent material like silica, and the mobile phase is passed down the column because of gravity. But in HPLC, a high-pressure pump is attached with the column. The high-pressure pump can generate a high pressure up to 40 megapascals. The column is filled with an absorbent material which has a very small particle size. The small particle size gives a very large surface area for the sample molecules to interact. As a result, the efficiency of separation increases, giving high resolution. Let's see the components of HPLC. The column is made up of stainless steel which can withstand a very high pressure up to 50 megapascals. The length of the column can vary from 5 to 25 centimeters and have an internal diameter of 4.5 millimeter. The flow rate of mobile phase through the column is usually 1 to 3 ml per minute. The stationary phase. As discussed earlier, the stationary phase is made up of an adsorbent material that has a very small particle size. The particle size is kept uniform to obtain a better performance. Usually, chemically modified silica, divinyl benzene, etc. are commonly used as a stationary phase. The mobile phase. The mixture of different solvents can be used as a mobile phase. The solvent used depends on the type of sample molecules which are to be separated. The mobile phase solvent can be polar or nonpolar. The mobile phase is usually kept in the solvent reservoir. The solvent reservoir is attached with a pump which pumps the mobile phase in the column with a high pressure. Just before HPLC column, there's an injector which allows introduction of the sample in the column. The detector. In order to detect the eluite, that is sample coming out of the column, the HPLC column is attached with a detector. Different types of detectors such as UV detector, IR detector, fluorescence detector, refractive index detector, mass spectrometer, electrochemical detectors, etc. can be used. The detectors are connected with a computer which collects the information. Now let's see the working of HPLC. As the sample molecules get separated, they are detected by the detector and a peak is obtained on the computer. This peak is plotted with respect to the retention time. To identify the components, we need to have standards. Let's understand this with an example. Let's say we run glucose as our sample and the peak of glucose is obtained at 5 minutes. Next, we run sucrose as our sample and the peak of sucrose is obtained at 8 minutes. Now we analyze the unknown sample for the detection of sugars present in it. Many peaks are obtained in the chromatogram. The peak obtained at 5 minutes indicates the presence of glucose. However, there is no peak at 8 minutes, which indicates that sucrose was absent in our sample. The remaining 2 peaks are still unknown as we don't have any standards to compare their retention time. This is how the detection of sample is done in HPLC. And for the detection, we must have the standards. Using HPLC, it is also possible to find the concentration of sample. But before doing so, we need to have standard curve. Let's say we take different concentration of glucose and run through HPLC. The peaks of glucose are obtained at 5 minutes. And with an increase in the concentration of glucose, the area under the curve increases. Once the standard information about the area under the curve and the concentration of glucose is obtained, just by measuring the area of glucose peak in the unknown sample, the concentration of the glucose can be estimated. Now let's see different types of HPLC. Normal phase HPLC. In this type of HPLC, the stationary phase is polar while the mobile phase is nonpolar. Usually, silica is preferred as a stationary phase as silica has SiOH group, which gives its polar nature. During normal phase HPLC, polar molecules are retained on the stationary phase, while the nonpolar molecules move fast down the column with the mobile phase. Reverse phase HPLC. In this method, the stationary phase is nonpolar while the mobile phase is polar. The nonpolar molecules are retained on the stationary phase while the polar molecules move fast down the column. Size exclusion HPLC. This method is based on size exclusion chromatography or gel permeation chromatography. In this method, the stationary phase particles are porous. As a result, small molecules gets inside the pores and takes a long time to move, while the large molecules easily pass down the column. Hence, separation occurs based on the molecular size. Ion exchange HPLC. In this method, the stationary phase has an ionic charge. If the molecules of interest has a positive charge, then a stationary phase is kept negative. And if the molecule of interest has a negative charge, then the stationary phase is kept positive. Hence, separation occurs based on the molecular charge. This method
Okay.
is based on size exclusion chromatography or gel permeation.
Okay, now you can you already understand the HPLC, the principle behind HPLC and the different types. But the most important is that the stationary phase, the selection of the stationary phase and the mobile phase, you need to know the physical chemical characteristic of your protein. If you don't know the protein and you are looking for separation and then further identification of the protein, this might need to, you need to test different stationary and mobile phase to detect the different proteins in your solution or your sample. So and selecting accordingly the type of HPLC you want to use, depending on the chemical, if you know it, if the chemical properties of your protein you are targeting. Otherwise, you can determine the sample and having different peaks, and then accordingly you can elute or separate the protein you want to study further from the HPLC separated solutions or components, and you further determine its chemical characteristics doing another experiment. So, but the HPLC is very efficient, and again, it's more advanced because it has a database already stored, and it can give you all these standard samples are already stored in the database, so it can give you according to the sample you're targeting, if it is protein, if it is carbohydrate, and so on. The other type of chromatographic techniques, as you can see, it's again this table gives, as it is already explained in the video, it's the same concept. There is a hydrophobic interaction. It's testing the hydrophobicity of your protein. For example, if you want to study protein, and this, for example, if you want to study high ionic or high charged proteins that have high for example, amino acids that have charged residues or charged side chains. So this one is, you can use it for hydrophobic interaction, or if you want to test for protein interaction, you need to study the protein and its type of hydrophobicity. There is an ion exchange, and this is for charge also residues in the case of proteins. There is affinity. If you want to test for the affinity between two proteins, then you can use affinity chromatography. You can use you can adapt the stationary phase to one type of protein and the mobile phase for another protein and test how they combine with each other. There is a dye affinity, of course, also it's another type. And this is for screening wide type of dye. It's not much used in protein interactions, but it can be used for proteins that can be tagged with probes or fluorescent dyes. There is the metal chelate also. It's another one. It's another type of chromatography that use the stationary phase that contains metals, or maybe use the mobile phase that contains metals. For example, if you are testing different protein interactions and that needs a mediator, metals, then you can test this using metals, chelates. There is the exclusion. This is excluding or separating the proteins according to their size. And this using the molecular size of the protein. But this one, it depends on the solubility again of the protein on the mobile phase, so that it can move on the stationary phase. So these are the different type of chromatography. It depends on your research question, your physical and chemical properties of your protein to select the specific method you want to use. Thank you. Do you have any question? Do you have any question for this session?
No question, Prof. Thank you so much for presenting.
Thank you. So please, I will upload today, hopefully, inshallah. I will upload today the three components or three documents for this session: the presentation, the study guide, and the record for today. Okay, I already recorded using the Google Meet. Okay, if not, then thank you for your presence, and see you next week then. And don't forget to send the assignment. Okay,
All right. Thank you so much, Prof.
bye. Have a nice day.